Hello everyone, myself Mahendhar Saratha. I'm a student and a research assistant at Grand Valley State University. Coming to this research, our goal was to make a biomedical literature more accessible and explorable, especially for people trying to understand how genes, diseases, chemicals, and pathways interact across research papers extracted from open source databases like NCBI and PubMed. We basically built a web application that converts these abstracts into interactive graphs, and these graphs usually represents relationships between genes, diseases, and pathways, allowing users to explore literature in a more intuitive way than traditional text-based search. Why this matters? So we already know biomedical literature is massive and constantly growing. It is difficult to understand and it is very time consuming to find connections manually. Tools today are mostly non-interactive and technical. So we want to solve above by making literature mining both visual and conversational. So coming to our system approach, we basically start querying our system based on the keyword mesh terms or else based on the research you want to interested in. Basically try to search something like genes related to cancer or to bone tissue cancer, and it will go to the PubMed. It will extract all the papers from the PubMed, and the PubData is then used to extract biomedical entities like genes, diseases from the text. We enrich this data with biological pathways using tools like KGRest and GProfiler. Relationships between the entities are stored in a graph database. We basically use Azure and Cosmos DB with Gremlin API support. Finally, we use D3JS to display these as an interactive graph. So let me share my application and let's discuss how to use it. Yeah, this is our application and this is our homepage. And if in case at any point of time, if you need help in using the application, you can always click on the help button and it will show how to use this application, and also there is a video to how to use the various options. And here you can search anything related to your research, something like genes related to bone tissue cancer. Gene pathways, you can always select minimum number of genes. Uh, For example, let's 3. To learn more about these fields, you can always click on the help button to understand. Click on the search. Let's wait for some time. And the D3.js will show us a visualization containing these gene pathways. Here you go. This is a visualization, like all the PubMed abstracts. These are the abstract. If you click on this dot, and you will be redirected to the PubMed abstract, and you can click on the pathway. You will be redirected to metadata for that pathway from KEG. And yeah, this is how it has given a visualization saying like all these papers are interacted using these gene pathways in common. And now we can use the GraphQL, and also we can use the open chart button to understand. Let's wait for some time so that the LLM will get trained on that data so that we can always ask the questions whatever we have about those related research. So coming here, we can always ask something like, okay, in a natural language, if you are expert in graph querying, you can always use this graph querying tab. If not, you can always use the natural language tab. Here, give me something like give me data for the knowing pathways, something like this, and it will give you data for signaling pathways, and you can see the updated visualization. And by using this, you can always explore more bigger and massive visualizations, something like uh, blood cancer, something like that. If you search for blood cancer, it will give you a huge amount of data with huge PubMed abstracts with relating with each other. In that case, you can always use this graph database to filter the data from the visualization, so you can explore more on the whatever the area of study you are interested in as a part of the research. And coming here, and yeah, this is how you're gonna use these tabs for graph querying. and there is always a summary button over here at the down. If you click on the summary button, it will generate a summary, small summary about that graph. It says like what are the kind of genes used, and also what are the kind of treatment plans used as part of research, and also what kind of future study they have suggested as part of those research papers. And let's wait for some time. It will generate a summary for us, and let me get into that after some time. Meanwhile, I'll explore this open chat button. Let's wait for some time. It will show up the open chat, which is basically a trained LLM. I think it's taking time for this LLM to show up. Let's wait for some time because it basically take all the data from PubMed and it will get trained on the data. It will basically take time. So here you go, graph summary. It's basically about the what are kind of collected articles. It will give a short photo file and description about those articles, and also the top treatments you have used as part of those papers, and also information about those treatments, and also kind of future work they have suggested as part of those papers. It's pretty cool. So you can always generate an updated visualization, click on the summary, and it will show you the summary to understand more about the visualization. Coming here, open chart. Okay, let's wait for some time. Yeah, before going into that, and you can always explore species, disease, and chemicals along. Yes, it's working now, and we'll open the chat, and it is a literature part. We basically trained all the data, whatever we extracted from PubMed, and trained that data with this LLM. And you can ask question like, "Hi, how many abstracts are used?" So it's basically gives how many abstracts that used in those uh, articles, like the article search. It's basically C total number of abstracts used is 570. For the search, whatever given, for this search, two genes related to two-bone tissue cancer, it has given the abstract for count as 570. That means it has extracted those 570 abstracts from PubMed, and it, and our application trained that data with the LLM so that LLM can answer whatever the queries you have about those abstracts, and also the graph query backed by graph database Azure Cosmos DB with Apache Gremlin support, and you can query the uh, graph as I shown before in a two ways, like GraphQL way, like you can basically enter the query, graph query, or else you can use a natural language option to, what give the in a more text and English way to and for the LLM to give the graph for whatever the way you like. Coming here, and you can ask questions like, "Hi, what are the top treatments used in those papers?" Something like this, and it will give you the answer. So whatever the treatments are used in those abstracts. So what are the published abstract? You can see it here. It has listed out treatments used in those papers. That's how you can use this literature part to ask your question regarding the study which is extracted from PubMed, and it will answer you straight away in more intuitive way. And along with that, graphs as well, and you can always click on the summary for the graph to understand more about the graph, and you can always use this tab to filter those graphs in a way you like. And so yeah, that's how we designed our interface and to search about, and you can always use the tell button to understand more about our tool. And but not that is not all we added for that, and it's powered by LLMs and also vector databases like Pinecone, allowing users to ask natural language queries. To wrap it up, so this project bridges LLMs, literature mining, and network biology into one platform. It can help researchers to quickly generate an hypothesis, identify diseases, mechanisms, or even explore drug repurposing opportunities, all in one user-friendly interface. And thank you everyone. And you can always use the link on our poster to understand more about our tool and research. Thank you so much.